Okay, biology folks, welcome back to Unit 6. And this is the second PowerPoint in Unit 6. And this is Chapter 17, and we're talking about protein synthesis, the making of proteins. And we talked in the previous chapters and units about the importance of DNA being the molecule of life and how important it is to pass that on to successive generations of cells. But we haven't really discussed like what is DNA is the recipe. What is the recipe for? What do we actually use that DNA for? And so hopefully this chapter will illuminate you on what we actually are using the DNA for. What it contains a recipe, what is a recipe for, and how do we get from recipe to the product? So think of having a recipe for really good chocolate chip cookies and what's going to be the process from the point that we get from the recipe for the chocolate chip cookies to actually a baked chocolate chip cookie. So it's kind of the same thing. We've got a recipe in the nuclei of our cells of the DNA, and it contains a recipe for proteins. And how does that actually translate into a protein? So that's what we're covering in Chapter 17. So it's pretty important information. So we're going to start with DNA, and we're going to end up with proteins, but we use this intermediate molecule called RNA, and we'll talk a little bit more about that. Going from DNA to proteins is a two-step process. The first step, going from DNA to RNA, is transcription, and then that RNA has to be translated into a protein. So the second step is translation. And if you recall way back a few chapters, what are the building blocks of proteins? The building blocks of proteins are amino acids. The bonds that hold those amino acids together are a special type of covalent bond called peptide bonds. And so once you get a bunch of amino acids together, it's called a polypeptide chain because there are lots of peptide bonds in there. And then eventually we're going to fold that polypeptide chain into a three-dimensional structure, and that will be the defining protein. So the relationship between genes and proteins is therefore one of information flow. So here's an overall picture, and this is found on page 339 of your books. And this is kind of an overall graphic description of what happens in protein synthesis. So you can see those two processes, transcription and translation. And you can see from this picture that this happens both in prokaryotes and eukaryotes. Slightly different in prokaryotes because you don't have a nucleus. Your DNA is just inside the cell, whereas in eukaryotes, part of the process is going to happen inside the nucleus and the other part of the process will happen outside the nucleus. So here's a real quick video for you to watch. It's a good YouTube video that you can use to get a picture of the overall process. There are also several videos posted in Canvas for you as well from the publisher. So we're going to start with this first step called transcription. And what we're going to do is we are going to convert DNA into RNA. This process occurs in the cell's nucleus. The DNA serves as a template for assembling a strand of RNA from the cell's free nucleotides. Remember when we talked about DNA replication, we said that the cell has free nucleotides. We have free nucleotides made of RNA as well. And so by using this process, evolutionarily speaking, we are able to leave the original DNA inside the nucleus for safekeeping. So if you think of DNA as being the hard drive of our bodies, Okay, you don't take your computer and leave it in a trunk in 90 or 100-degree heat. Um, you don't take your hard drive and spill coffee on it. So it's contains a lot of information and you want to keep it safe. It's the same thing with the DNA inside the nucleus of your cell. So here's what transcription looks like, and this is pictured on page 340 of your books. Okay, so you'll see there that the DNA strand is in blue, and that occurs inside the nucleus. Remember that it is just one side of the DNA strand. The complementary side would be on the other side. But like DNA replication, the DNA strand is going to unwind temporarily. And instead of making another DNA strand, we're going to make a strand of RNA. And it's a specific type of RNA called messenger RNA. And remember the base pairing rules for DNA? We said A always goes with T and C always goes with G. However, in RNA, there is no T, there is no thymine. So instead of thymine, the T, we have U. We substitute U in RNA as a general rule. And so what binds with A is going to be U. Okay, so you can see that we're going to make a strand of RNA based on one side of the DNA strand. That RNA is going to then leave the nucleus and it's going to go out to these ribosomes outside the nucleus, and then that RNA will be then translated into an amino acid chain or a polypeptide chain. And so here's just a different way of looking at it. Okay, you can see that this is undertaken by enzymes. That green blob in the middle is RNA polymerase, and that's going to kind of drive the making of RNA from one side of the DNA strand. The RNA in this case is the yellow strand. And here's another picture of that. Okay, so the big gray blob here is RNA polymerase. That's the enzyme. And the red strand is the RNA, and the two blue strands are the DNA, the original DNA. And this is pictured on page 344 of your books. So let's talk a little bit more about RNA. RNA stands for ribonucleic acid. And there are different types of RNA that we're going to discuss during this chapter. The first is called messenger RNA, or mRNA. And it is the only type of RNA to carry the protein building instructions. Okay, so this is the one that starts in the nucleus and then leaves the nucleus. So it's carrying a complementary copy of DNA out of the nucleus for the actual protein construction. We also have a type of RNA called ribosomal RNA, or rRNA, and it is the physical material that makes up the ribosomes. If you remember way back in the very beginning at the parts of the cell, we talked about endoplasmic reticulum and they had these purple dots on them. Those purple dots were ribosomes. Well, now we're going to delve into what actually happens at those purple dots, and those purple dots are actually made of ribosomal RNA. Then we're also going to talk about another type of RNA, the last type of RNA, called transfer RNA, or tRNA. And tRNA, as the name implies, delivers the amino acid to the ribosome. It transfers the amino acid to the ribosome. And that is going to happen in the order dictated by the messenger RNA. So the messenger RNA is carrying the message to the ribosomal RNA, the ribosome, and then in the order dictated by messenger RNA, transfer RNA is then going to transfer amino acids to the ribosome to make that protein. So let's talk about the chemical structure a little bit about DNA versus RNA. There are three main differences. The first is the sugar unit, the pentose, or the 5-carbon sugar. In DNA, the pentose is deoxyribose, and in RNA, the 5-carbon sugar is ribose. We talked a little bit about the base differences. The T is thymine in DNA, and it is U or uracil in RNA. There are no T's in RNA. And finally, you may have noticed just from the pictures, DNA is a double-stranded molecule, and RNA is a single-stranded molecule. So here's a picture of DNA versus RNA. And look on the left, okay, you'll see the T bonding with the A there on DNA. But over on the RNA, you'll see U's. You will not see any T's. Here's just a more detailed picture of RNA. And again, it's just a single strand, but it still has those sugar phosphate rotating sides like we see in the DNA. Okay, so let's get into the detail of transcription, the first part of protein synthesis. Let's first discuss this picture is located on page 343 of your books. And if you take a look, there's actually two pictures there. Figure 17.8 is a generalized picture for both prokaryotes and eukaryotes. And then the picture to the right of that, 17.9, is for specifically for eukaryotes. So one of the main differences between prokaryotes and eukaryotes is that prokaryotes only have one RNA polymerase. They only have one enzyme involved in this process, whereas eukaryotes have 3 different types of RNA polymerase, or 3 different types of enzymes. And there are 3 steps to transcription: initiation, elongation, and termination. And remember, the overall picture here is that we are converting the DNA to a single strand of messenger RNA to send that message outside of the nucleus. So when the RNA polymerase binds to the double-stranded DNA molecule, as soon as it binds, that causes the hydrogen bonds to break between the 2 DNA molecules, and the DNA unwinds. And so then what happens, this is specifically in eukaryotes, we have a region called the promoter, and it helps the RNA polymerase determine which strand is the coding strand. Remember, you've got 2 strands of DNA, and only one strand is going to be used. So RNA polymerase binds to the specific section of DNA bases called the promoter. Transcription factors bind to a box, a piece of DNA called TATA within the promoter section of DNA. And again, this is just in eukaryotes. So the RNA polymerase 2 is looking for a specific code called TATA. And once it attaches there, then it causes the DNA strand to unwind. And so this is pictured on page 343 of your books. This is the right-hand picture. And remember, this is just in eukaryotes. So the promoter is where the RNA polymerase binds. Okay, so then we get into step 2, the elongation of the RNA strand. And this is pictured on page 344 of your books. So the RNA polymerase moves along the DNA strand, unwinding the DNA and attaching RNA nucleotides. And remember, going from DNA to RNA, the G goes with C, C goes with G, T goes with A, on the DNA, but in RNA, the A goes with U. So if you take a look at the blue strands, okay, and by the way, this goes from the 5' to the 3' direction. If you take a look at that, you'll notice in the RNA polymerase bubble there, the blue strand is the DNA and the red strand is the messenger RNA. And you'll only see U's in the messenger RNA. You will not see T's. So the RNA strands are made in the 5' to 3' direction, and that's the same as we discussed in DNA replication. It happens in the same direction. After the RNA has passed, then the DNA strands, the two strands of DNA, rejoin, and those hydrogen bonds reform. So here's just going back to that picture on page 343 of your books showing the elongation. Then we go to the termination, step 3 of transcription. And again, this is different between prokaryotes and eukaryotes. In prokaryotes, the transcription stops after the polymerase passes the terminator sequence, and then the messenger RNA is released and finished. However, in eukaryotes, the transcription stops after polymerase II passes between 10 and 35 bases beyond what's called a polyadenylation sequence. The messenger RNA, referred to as pre-mRNA, is released, but then gets further processing before it leaves the nucleus. So the eukaryotes, they make the messenger RNA, but then it's going to get edited before it leaves the nucleus. And so here is that picture again on page 343 of your books, the overall steps of transcription, of making the messenger RNA. We again start with the initiation, the elongation, and the termination. So let's talk a little bit more specifically about this messenger RNA in eukaryotes. And this picture that I'm showing is on page 345 of your books. So before the messenger RNA leaves the nucleus, the ends of the messenger RNA are going to be modified or edited. The 5' end of the messenger RNA gets a 5' cap. The 3' end gets what's called a poly-A tail, and that is made of many adenine-based nucleotides. So it basically gets a bunch of A's added on to the 3' end. And so on the left-hand side of this picture, you can see the 5' cap. And then on the right-hand side, you can see that string of A's on the 3' tail. What are the reasons for this? We think that this aids in transporting the messenger RNA out of the nucleus. It helps maybe with guiding it. It helps protect the messenger RNA from being broken down by enzymes. If you recall, remember we had little aglets on the end of our chromosomes. Well, it seems like we have little aglets or little protective caps on the end of our messenger RNA before it leaves the nucleus. And this also helps the ribosomes attached to the 5' end of the messenger RNA to start translation. So that 5' cap helps with the messenger RNA binding to the ribosome. And again, this is pictured on page 345 of your books. However, in addition to that, there is additional, not only is there capping of the messenger RNA, but there is also some editing of the messenger RNA before it leaves the nucleus. And what happens there is that introns non-coding sections of messenger RNA are removed. And so what you end up with are called exons. And the way I remember this is that the exons part of the messenger RNA is what exits the nucleus. Okay, so introns are edited out and exons are spliced together, and that is what leaves the nucleus. So here's a picture of that. So here we have the exons and the introns, and we're seeing that the introns are being removed and the exons are then spliced together. And we have those yellow, the cap and the tail at the either end of the messenger RNA. And now the messenger RNA, now that it is edited, it is ready to leave the nucleus. Remember, because we're talking about leaving the nucleus, this only happens in eukaryotes. So we've made the messenger RNA, we've edited the messenger RNA, we've put a cap and tail on the messenger RNA. Now the messenger RNA leaves the nucleus and binds to the surface of the ribosomes. Now we have to talk about another term called codons. And codons are simply subdivisions of the messenger RNA. And what happens is once the messenger RNA is at the ribosome, the messenger RNA is read 3 letters at a time, and each 3 letters of the messenger RNA molecule is called a triplet or a codon. And we know that one codon, consisting of 3 letters of messenger RNA, ends up being 1 amino acid. Now, when we talk about transfer RNA, we're also going to talk about the anticodon. And the anticodon is the complementary sequence to the messenger RNA. So what is interesting, we're talking about the string of letters on the messenger RNA because those are the codons. Only 64 codons, regardless of what type of organism it is and on the entire Earth, there are only 64 codons identified. And what is even more interesting, if you look across all life on Earth, there are only 20 different amino acids. It's the order in which those amino acids occur that makes them different. We know that some codons serve as stop or start signals on the messenger RNA strand. And we also know that some different codons code for the same amino acid. So here's a good picture, and this is pictured on page 340 of your books. So again, we're looking at that DNA strand, and then we have the messenger RNA, and that shows the codons. Unfortunately, in this picture, it does not show the anticodons. We'll get to that in a little bit. And remember, the anticodons are on transfer RNA, but the codons are on messenger RNA. So we're now going to get to the second part of protein synthesis, and that's called translation. So we've transcribed the DNA into messenger RNA. Now we have to translate that messenger RNA into a protein. This also involves 3 steps: initiation, elongation, and termination. That's pretty convenient. It's the same 3 steps as in transcription. Okay, so let's go over those 3 steps. Initiation is where the messenger RNA and the transfer RNA are loaded onto the ribosome. So remember, we're out of the nucleus now, we're at the ribosome, the messenger RNA is loaded onto the ribosome. Elongation is as a new polypeptide chain is formed as that messenger RNA passes through the ribosomal subunits. And I like to picture it as kind of like the messenger RNA is a train that's driving through the tunnel, the purple ribosome tunnel. And then finally we have termination. That's where the stop codon is reached and the messenger RNA is released from the ribosome. And after the messenger RNA is released, the new polypeptide chain, the string of amino acids, are also going to be released. So let's talk a little bit more about that transfer RNA. We haven't discussed that very much. The transfer RNA brings the amino acid to the ribosome. It transfers the amino acid to the ribosome. It binds to the codons. Remember, the codons are the messenger RNA. So the transfer RNA binds to the messenger RNA and positions their amino acids in the order specified by the messenger RNA. Okay, so remember the messenger RNA is carrying the codons. The transfer RNA is carrying the anticodons. So again, the transfer RNA carries the anticodon to help bind to the codon. Now remember that the codon is a specific code and only certain anticodons can bind there. Actually, only one anticodon will match with the codon. So the transfer RNA serves to translate that messenger RNA into a specific sequence of amino acids. The ribosomal RNA, which is the material that makes up the ribosomes, helps build and assemble those amino acids into polypeptide chains. Okay, so let's get an even more detailed look at transfer RNA. And this is pictured on page 348 of your books. Okay, so it looks kind of like transfer RNA kind of looks like a lock and key. You can see at the bottom of the picture it has the anticodon, that three-letter sequence that's going to ultimately bind with the codon on messenger RNA. Okay, and so what they're using, instead of drawing that out on the left-hand side, that's the two-dimensional picture. The three-dimensional is in the middle, and then the very simplified version that they're using in this book looks like that upside-down teal sock. That's going to be the symbol for transfer RNA in this book. Okay, so the short sections of RNA made from DNA using the same translation process, the transfer RNA forms hydrogen bonds between their own bases to form a three-dimensional structure. The end that I talked about already, those green three letters on the bottom, carry the anticodon. The other end of the transfer RNA has an amino acid attachment site which bonds with one particular amino acid. So with that anticodon AAG, there's only one type of amino acid that will bind with that transfer RNA molecule. So again, we're talking about translation, and the first step is initiation. So the small ribosomal subunit attaches to messenger RNA. First, the transfer RNA attaches to the first messenger RNA codon. Now that first codon of messenger RNA, every protein that's made always starts with AUG. That's the codon that starts a new protein. The first transfer RNA always carries the first start amino acid. If you have a start, always have the same start codon, AUG, that AUG is always going to code for the amino acid called methionine. Then the second part of the ribosome attaches to the first ribosome. It's kind of divided into two. There's like a top half and a bottom half. Energy is needed for this to happen, and it comes from a molecule called GDP. And we're not going to spend a whole lot of time on that molecule, but just know that it it's helping with the translation process. So this is pictured on page 347 of your books. So let's take a look at this picture and dissect this picture a little bit. So the kind of salmon colored blob is the ribosome, and you'll see that it's kind of divided up into an upper part of the ribosome and a lower part of the ribosome. The messenger RNA is the orange single-stranded molecule that is kind of like driving through the tunnel, driving through the upper ribosome and lower ribosome. The messenger RNA is carrying the codons, 3 letters at a time. And then we have the kind of teal sock, which is the transfer RNA that is carrying the anticodon. And you'll notice the amino acid that's attached to the bottom of the sock or the top of the sock, depending on your perspective. Okay, so the purple things are amino acids. Once they're strung together, those amino acids become a polypeptide. Okay. And of course, it gets more complicated than that. So then we have step 2 of translation, which is the elongation of the amino acid chain. So what happens after the messenger RNA is loaded onto the ribosome? Then the transfer RNA binds to the messenger RNA codon. Remember, the transfer RNA is carrying the anticodon, and it binds to a specific area on the ribosome called the A site. And you can see that on the lower left picture called the A site. And this picture is found on page 347 of your books. Okay, so again, energy is supplied by a molecule called GTP, and that is used to make this process more efficient. So then the transfer RNA binds to the A site on the ribosome. And then you start having elongation of the amino acid chain, more transfer RNAs bind. Again, that is all dictated by the codons on the messenger RNA. And so then you get the amino acids being attached to the P site, and they are broken off of the transfer RNA. Once they are transferred in, then they break off of the transfer RNA. So you'll see here that the purple amino acids, which is a growing polypeptide chain, as they move through to the P site, then they get detached from the transfer RNA. And so energy from that GTP molecule is used to move the transfer RNA from the A site to the P site, and then again from the P site to the E site. And same thing going on there. The transfer RNA, then once it reaches the E site, the transfer RNA no longer is carrying the amino acid, and so it gets released. So the messenger RNA moves with its bonded transfer RNAs. And there's a video for you to watch this process. And there's kind of a video for you of the process. It's not a video, but you can click through the process there. And so here's just a different picture of the whole transfer RNA carrying the amino acid. This is pictured on page 349 of your books. So if you look at the transfer RNA, the teal sock carrying the anticodon, and how it binds, that gray is actually the amino acid tyrosine, and how that whole process works. Okay, and you can get a look at the computer model, kind of the three-dimensional model there on the lower right. And so here again is kind of the overall process. And this is pictured on page 353 of your books. There is a much more detailed, page 353 of your books is a much more detailed picture. I kind of gave you that picture on the previous slide, but if you want to work through it, there are more steps pictured on page 353 of your books. So here's just kind of a different look at it. These are kind of just some different graphics of the same process to give you an idea. So in this case, the ribosome is blue and the messenger RNA is red or pink. And then in this case, the transfer RNA is green. And in the middle, it shows the codon in red and the anticodon in green. You can see that they are complementary. And then this kind of shows the A and the P site, but it does not show the E site. So it's just kind of a simplified version of the other graphics that we've been seeing. And again, here's that three-dimensional graphic that I showed you on page 349. Now, just as soon as you think you understand it, things get complicated, and you probably never realize that all this is happening all the time in your cells. But does this happen only one ribosome at a time? No. One strand of messenger RNA actually may be read by many ribosomes, all making the same protein at the same time. So what it does is that there may be a string of ribosomes forming a long tunnel for that messenger RNA, and after at least one ribosome, it'll go to the next ribosome. Now, it's carrying the same code, it's going to make the same protein, but your body may need a bunch of the same protein. So you'll have a string of ribosomes, and that one messenger RNA molecule will travel through all those ribosomes to make the same protein. And this is talked about on page 355 of your books. And so here is the BioFlix representation or video on protein synthesis. Also in Canvas, it has the transcription and translation in more detail, 2 separate videos that I've loaded in Canvas for you. I just didn't embed it in this PowerPoint. So make sure you watch those videos as well. It will help you visualize these processes. So let's go back to the DNA and talk about mutations. We talked a little bit about mutations and what can happen when something happens, especially during cell division. So we talked about some of those problems. So we can say that one letter is substituted for another. Okay, maybe we have an A going with a C instead of the T in the DNA. Or we just can have a random letter inserted. Or we can have a missing letter. Remember, mutation always happens at the DNA level. It happens at the DNA level. Or like that Tata box that we were talking about, maybe there's 3 copies of that instead of 1. So there could be gene duplication. So here is a nice chart for you to look at. And this talks about the different types of mutations and what can happen as a result of those mutations. And actually, when you take Bio 2, you'll talk a little bit more about this. So, a substitution, you can have one DNA base being substituted for another in the DNA, or we talked about an insertion or deletion. So if we have a substitution of one DNA base for another, um, one possibility is that it's silent. It doesn't result in a change of amino acids. Remember that there's only 20 amino acids, but there's 64 codons. So it might be a change that results in the same amino acid. We can have a missense in a substitution mutation, and that's where there's one amino acid substituted for another. And you can have a nonsense mutation that changes an amino acid to a stop codon. And so that's just going to cut short the protein. Now, another type of mutation that we talked about are insertion or deletions into the DNA. And then if you insert or delete one letter into all those codons, you can see that that's going to shift all those codons. So that will alter the grouping of the codons, and then that's going to change the whole protein. It's going to change all the amino acids from the point of that mutation on, and it's just going to change everything from that point on. So, what are things that cause mutations? Those are called mutagens. We know of certain things that are mutagenic. We know that cigarette smoke mutates a particular gene on your DNA. We know that ultraviolet light mutates your DNA. And we also know that radiation mutates your DNA. Okay, so we know all 3 of those substances mutate your DNA. Now remember, we talked in the last unit that to some extent your DNA has the ability to repair itself. And in fact, if you go out in the sun and you're mutating your DNA will go, okay, I can fix this, and it will fix it and fix it. But after a while, if you keep doing it, then eventually it breaks down. It says, I give up, and you get skin cancer. So again, we know that cigarette smoke mutates your DNA. Ultraviolet light mutates your DNA. Radiation mutates your DNA. That's why the dental technician leaves the room. And that's also why you wear the lead shield. And here are some of the effects of mutations. Now you won't see these vegetables for sale in Publix, but they still taste the same. And here's a poor fruit fly that has mutated DNA for his eye. And so his normal fruit fly is on the left, and then the guy with a mutated DNA is on the right. So what happens if a mutation occurs? It can be harmful. It could be beneficial. Okay. I would suspect that some folks that are in the Olympics or play for NFL, that their DNA is different from mine that enables them to excel at the sport, and I can't do that. So some mutations may be beneficial. Some may be neutral. You may not even know that it's a mutation. So how can this affect it? We talked a bit about this. It will affect the protein synthesis. We can replace one amino acid with another, or we can get an entirely different protein. So here's a picture from page 342 of your books. And if you take a look, some folks have been playing with genes. On the left is a picture of a tobacco plant where they have inserted the gene from fireflies that make them glow. Can you imagine driving down a rural road at night and seeing this field of tobacco plants that's glowing in the dark? And then on the right, they have taken the gene from a jellyfish that makes it glow and put it on the poor piggy's snout and hooves. And so now he glows in the dark. So you can read about this on page 342 of your books. So this concludes chapter 17 on protein synthesis. Again, I want you to spend some time with the material and be able to understand the process, how we're getting from DNA to a three-dimensional protein. Remember, DNA is this linear molecule. It's a string of letters, but now we have to get to a three-dimensional protein. So as usual, let me know if you have any questions, and next up will be chapter 18 on gene expression.